Chapter 5 is about microbial metabolism, and this is your list of study objectives for this chapter. So, we're going to start with a discussion of metabolism. Metabolism is defined as the totality of all the controlled biochemical reactions that take place within the cell, for our purposes within the microbe. And the ultimate purpose or function of metabolism is reproduction, to reproduce the organism. So put a star by that. The ultimate function or purpose of metabolism is to reproduce the organism. So all the chemical reactions, which are all catalyzed by enzymes, add up to a totality of all the reactions in the cell being the metabolism of the cell. Now you can break down the metabolism into two basic categories: catabolism, so catabolic pathways or breakdown pathways that release energy, and anabolic pathways, anabolism, which are synthetic processes or synthetic reactions that require energy. And all of these together add up to the metabolism, and the purpose of the metabolism is reproduction of the organism. You can have a cell that's alive, but if it doesn't divide and reproduce, then it's not going to pass on its genetic material to the next generation. It's not going to, in our case, cause disease, because one cell by itself doesn't cause disease. It's a huge number of cells of the same type that can cause it. So an infection, isn't caused by one cell. It's caused by one cell that divides and divides and divides and creates a lot of cells that cause the problem. So reproduction is critical to pathogenesis, and that comes down to the metabolism of the cell. All right, so we have some fundamental statements that are true of all organisms. We're focusing on pathogens, we're focusing on microscopic organisms in this course, but these are true of all organisms in biology. Every cell has to get nutrients, molecules, biomolecules, from the environment, from outside the cell. Energy ultimately comes from either sunlight, or sunlight is converted into glucose by photosynthesis, and then catabolism of glucose provides energy. But ultimately, either sunlight, directly from sunlight, or from the catabolism of biomolecules that contain the energy of sunlight. The immediate form of energy that a cell wants to use is adenosine triphosphate, ATP. Although I should add that GTP is also an important form of energy in the cell, guanosine triphosphate. Cells catabolize nutrients into monomers, so that's usually called it's called hydrolysis. So a breakdown of a polymer into monomers, but then monomers that are glucose can be broken down or oxidized to make ATP. And so monomers from nutrients, energy from ATP, and enzymes are all important in the anabolic reactions of the cell. Cells assemble macromolecules into cellular structures. Cells can only reproduce once they have built enough structures to sustain two daughter cells. This picture is crazy, but if you really look at it a little bit at a time, it's describing all of the metabolism of a cell that you probably learned throughout a large portion of your biology 1406 course. So this represents kind of any cell. And nutrients have to come in. Those can be amino acids, they can be full-on proteins, they can be carbohydrates, fats, they can be monomers or polymers. And what has to happen in the cell is some of those nutrients are going to get broken down. So here what we're really talking about is the breakdown all the way to carbon dioxide, so respiration. So catabolism is respiration, breakdown of glucose. And that transfers some of that energy into ATP. ATP is used to build other things like macromolecules and then organelles, so the cell can increase in size and eventually divide. So here's the cell division here at the bottom center of this image. And if you don't have nutrients coming in that are bringing in not only chemical material but also energy, then this whole process would stop. Or if any of the enzymes that are doing all of this work aren't working, then this whole thing would stop. So the enzymes have to work together. The nutrients have to keep coming in. Oxygen typically has to come in. So there are a lot of things that keep this process going. And this process is the metabolism that ends in cell division. Now, just a review of some things you should know from 1406, and if not, refresh yourself. This chart shows on the y-axis energy, free energy, of any of the reactants in a particular reaction. And on the right side at the bottom, you see the individual products. So in this example, this is a breakdown reaction where something larger is broken down into its component parts. All reactions have what we call an activation energy, a certain requirement that if a certain amount of energy is not put in or invested into the reaction cannot move forward. Most reactions by themselves cannot get enough energy to get over that barrier. But an enzyme acts to lower the barrier and therefore allows the reaction to move forward. And what enzymes do is they push or pull the reactants together or apart to help make the reaction happen much faster. In this image, you see an enzyme shown primarily in blue. And this image goes into some detail that you probably did not see in 1406, but you now need to know for microbiology. An enzyme actually has several parts. The protein portion, if it's a protein enzyme, which a lot of them are, is shown in blue. That's called the apoenzyme, the protein part. But a lot of proteins need to also have attached to that protein part some kind of cofactor and or some kind of coenzyme. So the cofactor is shown in orange. It can be, they're just showing something bound there. And the coenzyme is shown in red. Only when all of the required parts are together, then you have what's called the holoenzyme. Only the holoenzyme is able to catalyze the reaction. Now, cofactors and coenzymes are really vitamins and minerals. So your vitamins and your minerals are required to make your enzymes, your apoenzymes, functional. So only if all of those parts are there do you have the holoenzyme, which is the actual active enzyme. Now, you'll remember that in this picture, they're just kind of showing the enzyme. Of course, this would be the holoenzyme, but they're not showing the different colors this time. But this is the holoenzyme. The substrate fits into the active site, and then the active site changes shape a little bit, or the enzyme changes shape a little bit to actually make it fit even better. That's called induced fit. And the combination of the substrate with the enzyme is called the enzyme-substrate complex. Now, how does an enzyme work? The substrate binds to the active site. You have the enzyme-substrate complex, like I said. The enzyme changes shape to make it fit even better, as you can see here. It really doesn't matter particularly what reaction is being shown here. They're just really using one example to show you what's happening. And then the enzyme pushes or pulls or otherwise affects the substrate, in this case breaks it apart and releases the products. So the products are shown here. And then the enzyme returns back to its beginning state and it can take another substrate. So the enzyme is not destroyed or used up as a part of the cycle. And that's why it's a cycle, because you start with an enzyme and you end with an enzyme, and anything that's named a cycle has to start with and end with something. So in this case, the enzyme stays in the cycle. Now, the substrate is converted to product, and then when the cycle goes again, a different substrate is converted to product. But the enzyme itself can repeat over and over again through the catalytic cycle. So especially for protein enzymes, but also for RNA enzymes, there are factors that can influence the rate. The rate is the speed in which they do their catalytic cycle, so how fast they can produce each product. And how fast enzymes can produce their products governs how fast the metabolism of the cell is. And you'll remember we said the metabolism of the cell is all about reproduction. So if the enzymes are able to do everything quickly, then the cell can probably divide more quickly. If the enzymes are working more slowly, the cell will end up dividing. It'll take longer for the cell to divide, I should say. So if we're talking about a pathogen, like a bacterial infection, then what we're talking about is how fast the enzymes work is going to determine how quick the infection is going to spread. And that's really important, because if an infection spreads really quickly, it can overtake the immune system in the body and be very bad. If an infection moves very slowly, then the immune system will be able to fight it off much more effectively. So this actually, in the end, ends up mattering a whole lot in terms of how bad an infection is and how the patient responds to it and fights it off. So there are factors that affect the enzyme's temperature, pH, the concentration of the enzyme itself, how much is there, how much substrate is there, and the presence of inhibitor molecules. So let's talk about temperature. When a protein enzyme is folded, the image on the left is showing a fully folded functional protein. Excuse me. And on the right side, you see a denatured protein. So it's unfolded. It doesn't have its three-dimensional shape. And the things that cause proteins to denature are high heat, extreme pH, and also salt. A lot of salt can cause a protein to denature. So high heat, extreme pH, and extreme pH means very basic or very acidic. And then salt. Low temperatures do not denature proteins. Low temperatures just make the proteins move more slowly. The cycle of the enzyme will be slower, but it won't denature the proteins. That's really important. So low temperatures, like freezing something, does not denature the proteins. Now, inhibitors will attach to the active site, or they can attach to somewhere other than the active site. In this picture, we have what we call a competitive inhibitor. If it's competitive, it will actually bind to the active site, and what you can see here is that causes the real substrate not to be able to bind. There are also inhibitors that can bind somewhere else on the enzyme, and then it causes a change in the shape of the active site, but then the outcome is still the same. The substrate can no longer bind there. Inhibitors are important for when we think about creating drugs that stop the metabolism of a pathogen. What we're really thinking about is chemicals that act as inhibitors to the pathogen's enzymes. Because if you can stop the enzymes of the pathogen from being able to catalyze reactions, you can stop the metabolism of the pathogen itself, and you can stop the reproduction of the pathogen. So competitive inhibitors, well, even non-competitive inhibitors, enzyme inhibitors are drugs. They are the drugs that we find, that we figure out, that can block the metabolism of the pathogen's enzymes.